Our discussion over the last several lectures has focused on the transmission of physiological information among the parts of a complex multicellular organism. We've seen that animals have evolved exquisitely elaborate mechanisms for long-distance chemical and electrical communication, systems that we call the endocrine and nervous systems, respectively. Let me remind you, however, of the broader context in which we've discussed these mechanisms, which has to do with an organism's ability to coordinate its various parts in order for the organism to function as a whole and specifically to be able to homeostatically regulate its internal environment. But there are a couple of other important aspects of how organisms, animals in specific, maintain themselves in an adaptive fashion that we have yet to address. These have to do with the fact that animals have to interact actively with their external environment as well. Consider the following scene. Moths typically fly at night. This is unlike their butterfly cousins, which typically fly during the day. And if you were out at night and you could see moths, even though it was dark, you'd mostly see male moths flying around searching for receptive female moths to mate with. Now, how do the males find the females, especially in the pitch dark? Well, it turns out that the females release chemical signals called pheromones. The males have special receptors on their antennae that detect these chemical signals, and they use this to follow a trail, essentially upwind, following a plume until they find the female. Now, at the same time that the male moths are trying to find female moths, bats are trying to find moths to eat them. How do the bats find the moths in the dark? Well, as you may know, bats produce extremely high-pitched sounds that they use as a kind of sonar. We call these echolocation signals. And this sonar system allows the bats, even in the pitch black, to detect moths, guiding them in and allowing them to capture prey. Fortunately for some species of moths, moths actually have a specialized ear that detects the bat's echolocation cries, and if a moth hears a bat, it dives for the ground very rapidly where it's going to be safe. Now, this nightly drama illustrates the next two issues that we need to address in this lecture and the following. The first is to ask how information, especially information from the external environment, is detected and converted into a physiological signal that the organism can use. The male moth needs to get information about the presence of a female, which it does by detecting a chemical signal. The bat needs to gain information about the presence of the moth, which it does by detecting an acoustic signal. This kind of question will lead us today to consider how sensory systems work. Now, the second issue that we need to address is to ask how animals respond in an adaptive fashion to this kind of information. When the male moth detects a female, it flies to the female. When it detects a bat, it flies to the ground. If the bat detects a male moth, it flies towards the moth. Fundamental to any of these adaptive responses, obviously, is an animal's ability to move. And this will lead us to look in the next lecture at the mechanisms that enable movement in animals. Okay, so let's begin today with an overview of what I mean by a sensory system. Sensory systems gather information from the environment through the activity of specialized cells called sensory receptors or sensory receptor cells that are specialized to detect particular kinds of physical stimuli and to convert those physical stimuli to physiological signals so that it can be transmitted to other parts of the organism. Sensory receptors are often assembled with other types of cells into something that we would call a sensory organ. Sensory organs are structures that enhance the ability of sensory cells to collect information and even in some cases to start processing that information. The most obvious sensory organs that we think of are things like eyes and ears and so forth. Now, although the example I began this lecture with emphasized the acquisition of information from the external environment, let's not forget that we also need information from the internal environment as well, and there are sensory receptors specialized for this task. We've already seen examples of this when we first talked about homeostasis. For example, cells in the mammalian hypothalamus, you'll recall, are specialized to detect temperature. And they use this information to help maintain the normal body temperature of the animal, as an example of an internal sensory system. There are many, many other kinds of internal sensory receptors detecting things about, for example, the chemistry of our blood, the positions of our joints and muscles, the status of our digestive tracts, and so forth. Now, In our conscious experience, we usually think of sensory systems as detecting information about the outside, providing the basis for the perceptual experiences of what we would call sensations, things like sight and sound and touch or taste and smell. Actually, you know this. There's an enormous range of the kinds of physical information that any one species, ourselves included, but anything, might be able to detect from the environment. Even for ourselves, you can imagine all of the different kinds of rich sensory experiences we have. But then if you include other organisms like bats and moths or whatever, then you have an enormous range of kinds of information that might be gathered from the environment that could be useful to the organism. Given this diversity, it might seem like it's a daunting task to begin to understand how sensory systems can work in general. But fortunately for us, in spite of this enormous diversity, all sensory systems share a few basic properties in the way they detect and encode information. And by understanding the mechanisms that underlie these basic properties, we can gain an appreciation of how sensory systems work in general, even if we're not going to be able to cover all the details. Now, for the sake of time, we're going to focus our discussion today around one of the most fundamental properties of sensory perception, which we refer to as the modality of a sensation. Now, what do I mean by modality? Different forms of physical stimuli, such as light or heat or chemicals and so forth, are transformed by sensory systems into distinctly different qualities of sensation. And we refer to these different qualities as perceptual modalities. In our own perceptual world, for example, these modalities refer to different sensations that we would refer to, say, as vision on the one hand, or smell on the other, or touch, and so forth. Of course, we could define modalities on a much finer scale, and we will a little bit at the end of the lecture. And of course, as I said, other animals use other sensory modalities as well. But the point here is that we have to begin to account for the physiological mechanisms that are responsible for coding such distinct sensory modalities to give us such distinct sensations. And that's where we're going to begin. Now, actually, it was Aristotle, the Greek philosopher, biologist, who we've run into a number of times before in this course, who first proposed that you could divide sensation into what he called the 5 primary sensory energies, and he defined those as sight, sound, touch, smell, and taste. And in fact, we would still define these as the major sensations that we have in our own human experience. Now, our modern understanding of the physiological underpinnings that are responsible for encoding these kinds of different sensations, actually, I would trace back to the 1830s in the publication of a textbook called Handbook of Physiology by a German anatomist and physiologist named Johannes Müller. Now, Müller was one of the leading biologists of his time. And his Handbook of Physiology actually served as the definitive text in this area throughout Europe for a decade or more. What Müller proposed is the following. He suggested what we now call the law of specific sensory energies. And this law stated that there are different kinds of sensory nerves, and that each kind of sensory nerve carries its own distinct, what he called specific sensory energy. Thus, Müller argued, we perceive light when sensory nerves that are explicitly associated with vision are stimulated, because the sensation of light is defined by the sensory energy that these nerves carry. Now, Müller actually almost had it completely right, as it turns out. As he suggested, the quality or modality of a sensation that we perceive results from, or I should say, is a property of the nerve itself, not a consequence necessarily of the physical stimulus that excites that nerve. For example, if you're given a sharp blow to the eye, I mean, God forbid, but if somebody punches you in the eye, you'll actually see a flash of light. Now, this is not because the eye is specialized to convert that physical blow into some sort of visual sensation. It's just that these sensory neurons in your eye, if they're physically deformed enough by a sharp blow, will actually be stimulated. And you perceive that stimulation as light, even though the initial stimulus had nothing to do with light. In fact, there's a very interesting story about a court case that Muller was called to serve as an expert witness. Apparently, someone had been attacked in the pitch black, and he claimed that he could identify his assailant because when he was attacked, he was punched in the eye, and there was a flash of light, and the claimant said that he could then, in this flash of light, see who had attacked him. Now, Muller pointed out that although he was sure that the claimant did in fact experience a flash of light, there was no way he could have seen his assailant because the sensation was an entirely internal phenomenon, a misplaced sensation caused by the specific sensory energy of the sensory cells of his visual system being stimulated by the blow. Now, Muller was correct that each kind of sensory nerve only carries information about a particular sensory modality, but his theory wasn't all that clear about the mechanism by which this specificity was achieved. In fact, we now know that all sensory neurons, in fact, like all neurons, respond to stimulation in only one way. If we put electrodes in those neurons, the only thing they do is fire action potential. That's what neurons do. That's what sensory cells do. So it isn't as though each cell carries a specific energy that's unique to that cell. The cell fires or it doesn't. But the specificity of sensory modalities is achieved in a way that is similar to what Muller proposed. In fact, there are two components to this. The first part is what Muller got exactly right, and that is that the specificity of sensory modality depends on anatomy. Sensory neurons, or the neurons that they connect to if there's several cells in the line, send their information to only one particular area of the nervous system, an area that's specialized for receiving and processing only one kind of sensory information. So, for example, if you stimulate cells, sensory cells in the eye with a physical blow, that information is sent to visual processing systems, mostly in the back of your brain, and those visual processing systems interpret those action potentials they receive as something having to do with light. It's really just a matter of wiring, regardless of how those sensory cells are stimulated. Now, a second point, one that Muller wasn't actually able to get at, was that, although he actually suggested it must work like this, was that different kinds of sensory neurons actually will respond best to only one physical stimulus. Think back to Muller's court testimony of someone being struck in the eye with a blow. Now, the sensory cells associated with vision in the eye can be stimulated by pressure if the pressure is great enough, but normally they're not. They are instead exquisitely sensitive to light stimuli. And that's what they're specialized to receive. So this sensory specialization that gives us a sensation of different modalities has a lot to do with how those cells actually are specialized to convert particular kinds of physical stimuli. Now, what's interesting to me is that there are actually only a very limited set of mechanisms by which physical stimuli are converted into neural physiological signals. And it's this topic that we're going to spend just a little bit of time on next. Let's remind ourselves of what a sensory receptor has to accomplish, though, first. Remember, a sensory receptor has to be able to convert a physical stimulus into some sort of physiological signal, a neural signal, ultimately action potentials. And this conversion, we know from what we've been talking about over the last few lectures, has to somehow involve a change in the membrane potential of that sensory receptor cell, because it's a change in the electrical potential of the cell, which ultimately is going to lead to the generation of action potentials down the line. So a sensory receptor cell then must have a mechanism by which some physical stimulus causes gated ion channels to open or close. This process of converting a signal from one form to another, one form of energy to another, is generally called transduction, and we refer to it as sensory transduction in the case of receptor cells. So the question then is, how does a sensory receptor cell convert a physical stimulus into a change in membrane potential? And as I said, despite the enormous diversity of kinds of sensory receptors found in all sorts of animal sensory systems, there are really only 5 basic mechanisms. These are what we call chemoreception, mechanoreception, photoreception, thermoreception, and electroreception. Each of these transduction mechanisms converts involves a physical sensory stimulus influencing the activity of some kind of gated ion channel. Let's look at how this works briefly in each case. First, let's consider chemoreception, which we're actually kind of familiar with at this point, because chemoreceptors function much the same way as the receptors that we've talked about before in the context of cellular signaling or neurotransmitter signaling, a number of examples. The molecule that's being detected, the physical stimulus, simply binds selectively to a membrane-bound protein that serves as a receptor, initiating some sort of second messenger cascade that in turn will cause ion channels to open and close. This mechanism is really no different in its basic design from any of the other kinds of cell signaling receptor mechanisms we've looked at. Now, chemoreceptors are very ubiquitous in our own sensory experience. Obviously, they provide the basis of both taste and smell. But many other sensory processes involve chemical signals as well. Remember in that example I gave with the moth, the male moth, was using a chemoreceptor to detect the female's pheromone. In this case, the moth's antenna has a receptor that is very specific for binding only the chemical compound that the female releases as a pheromone. Other organisms use chemical signals in a variety of ways, for example, to identify relatives which may smell different, or even group members that develop different smells, or to mark defended territories and so forth. In fact, chemoreception is probably the most evolutionary ancient form of sensory reception, and certainly the most ubiquitous form of sensory reception in the functioning of organisms. Now, a second type of transduction mechanism is mechanoreception. Mechanoreceptors are cells that are specialized to detect mechanical forces that distort their cell membranes. Now, the way that mechanoreceptors work is conceptually very simple. In fact, it's remarkably simple. It appears as though mechanoreceptive cells have ion channels. Remember, these are proteins that span the membrane that are very tightly bound to other proteins serving as structural supports on both the inside and the outside of a cell membrane. So essentially, these channels are very tightly bound to a scaffolding. If the cell membrane is distorted, I mean, literally by something pressing on the surface of the organism, then mechanoreceptors convert this into a physiological stimulus, because this distortion of cell membranes literally pulls apart these channels, literally forcing open ion channels, because the distortion and the connection to this scaffolding causes these proteins to actually get stretched. And that's the basis of mechanoreception. Now, the details of mechanoreception on a molecular level are actually still quite not fully understood, but the basic mechanism seems to be that simple. In our own sensory world, mechanoreceptors obviously form the basis of our sense of touch. There are many, many different kinds of mechanoreceptors in our skin, each specialized to deal with different kinds of touch in terms of the strength of the sensation, the nature of how the sensation changes through time. Mechanoreceptors also form the basis of our sense of hearing. Now, it turns out that the way the ear works, I won't go into this in any detail, is that the inner part of your ear actually has a membrane that is covered with cells that have small hair-like structures sticking up. And when this membrane is distorted, which it is when sound, the air pressure changes that are sound, impinges on them, when this membrane is distorted, the distortion of this tissue layer actually causes these hairs in these hair cells to bend. And the bending of the hair cells causes mechanoreceptors in those hair cells to open up, causing those cells to depolarize and fire action potentials. Now, a third kind of transduction mechanism is what we call photoreception. Photoreception is a bit more complicated, but we can simplify it just a little bit by looking at the essence of it. Photoreceptors include a pigment called rhodopsin that can absorb photons of light, and when it absorbs these photons, that added energy causes the rhodopsin pigment to change its shape. Actually, rhodopsin is made up of a couple of units, a protein called opsin, and then another molecule called retinal. And the retinal is derived from vitamin A, which is why vitamin A is good for vision, we're told. It's the retinal molecule that actually allows the complex to absorb the light. And then the opsin, when it changes its shape in response to this, will, through that shape change, as we've seen many times before, trigger a second messenger cascade, which will then cause ion channels to open or close. In this way, electromagnetic radiation, such as photons of light, is transduced into a change in the membrane potential of the photoreceptor through this second messenger cascade originally triggered by a change in the rhodopsin molecule. We're going to come back to light in just a second. Now, thermoreception is another fundamental sensory transduction mechanism, but one that's less well understood, so I'll only mention it briefly here. Thermoreception, of course, gives us information about internal and external temperature and so forth. Thermoreceptors are less well understood. It seems as though there are separate receptor mechanisms for detecting extreme heat and extreme cold, but in both cases it seems as though ion channels are associated with a temperature-sensitive enzyme in the membrane. This enzyme changes its activity as the temperature changes, which in turn changes the activity of the ion channel. Now, there's one last fundamental mechanism of sensory transduction that I'll just mention, and this is electroreception. We know least about this because actually very few organisms have electroreceptors. We don't, as far as we know. The mechanism must be similar to whatever the mechanisms we talked about when we were talking about voltage-gated ion channels when we talked about the action potential, and that is a change in electric potential across the membrane must change the shape of a protein. Electroreception provides an excellent example of how the sensory worlds of different kinds of animals can be so different from our own. For example, some species of fish, such as catfish, are actually very good at detecting weak electric fields, things we're not even aware of. They use this to find prey because their prey will, as living organisms do, generate a weak field around them. And the catfish will, in the complete dark, find its prey by looking at distortions in the electric field. There are other kinds of fish, these are called the electric fish, both in Africa and South America, that have actually also generated organs that can distort the electric field very strongly. And they actually use these distortion organs, these basically electro-generating organisms, to create communication signals. So they're actually sending signals between each other in the pitch black in this modality, which is basically an electrical modality that we're not even aware of. Now, what's remarkable to me is that as far as we know, all sensory transduction really relies on one of these five basic mechanisms. And in fact, just those first three, chemoreception, mechanoreception, and photoreception, seem to account for the majority of it. To repeat a point I made a couple of lectures ago at the end, it really all boils down to the opening and closing of ion channels, getting this whole sensory reception process started. Of course, the modality of a sensation is really only the broadest characteristic of a perceptual stimulus that we might want to consider. When you think about our own sensory experience of vision or sound and so forth, it's obvious that much, much more information has to be captured and encoded and processed by sensory systems to create the very complex sensations that we're aware of. And there's a lot known about that, but what I want to do is to just take one little next step, and that is to ask, how can sensory reception within any of these modalities be more finely tuned to a narrow range of stimulus frequencies? Consider our perception of sound, for example. We're aware not only of a sound occurring or not, but we're also aware of the frequency of that sound, whether it's a high-pitched sound or a low-pitched sound. We're able to discriminate, among a range of frequencies, and so our receptive system must be tuned in some way. How does this kind of further specificity arise? Well, in some cases it's easy to imagine. For example, in chemoreception, the specificity is inherent in the mechanism itself. A particular kind of molecule will bind to a particular kind of receptor, and so it's inherently a highly specific system. In the case of sound, it's not so clear. In fact, all mechanoreceptors, all hair cells in the ear, respond the same way to different frequencies of sound. But in that case, it turns out that the receptive organ, the sensory organ itself, is what causes this differential tuning. So the tissue sheet in which these hair cells are located in the inner ear changes its properties, both its width and its stiffness, along a length. And along this length, different frequencies of sounds will cause a maximum distortion. So a low frequency sound will cause one part of this tissue to bend more, and a high frequency sound will cause the other part of the tissue to bend more. The receptor cells will just fire or not, but the ones that are located in the part that bends more are going to be the ones that are firing, and so it's actually the position of these receptor cells all acting the same way where the tuning of our sense of sound comes from. And again, this is wired up specifically to parts of the brain that are processing these different frequencies. Now, vision provides a particularly interesting case of how receptors can be tuned to a narrow range of frequencies, or what in the visual modality we call different wavelengths of light. I want to give you a little background on this by telling you about John Dalton, who was a very celebrated physical chemist working in the late 18th century and early 19th century. It turns out that Dalton realized when he was very young that he saw color differently from other people. Now, he was, as we would say today, colorblind. The interesting thing is that Dalton developed a very specific hypothesis for why he would be colorblind. He suggested that the reason that he couldn't see certain colors, which gave him his different sensory perception, was because the fluid in his eyeballs must be colored differently. In fact, he suggested the fluid must be blue. And the blue fluid would then filter out certain wavelengths of light, allowing him not to see those wavelengths and thus changing his sensory perception. The interesting thing about this is that Dalton actually wanted to test this hypothesis, and so he willed that his eyes be dissected when he died and that the fluid in them be examined to test this hypothesis. Well, Dalton died many years later at 78 in 1844. His eyes were dissected out, and when the people who did the dissection looked, they saw that the fluid in his eyes was the same as in anybody else's eyes. So that hypothesis was clearly wrong. In fact, they even cut the backs off the eyes so that they could look through them. And when they did that, they didn't see any color difference. Well, we know now that what causes color blindness are mutations in different kinds of opsins. Right? That's the protein component of rhodopsin. Different kinds of opsins that are specialized for best receiving different wavelengths of light. In fact, in human vision, there are 3 major kinds of opsins that are found in different sensory cells that are specific for colors, cells we call cones. Cells that are tuned specifically to blue light, to green light, and to red light. And what we know is that many colorblind people suffer from color blindness because of a mutation in one of these opsins that makes that opsin non-functional, so they can only see 1 or 2 of the total 3 colors that we would normally be able to perceive. Now, of course, we see more colors than 3, and that's because the information from these 3 basic color cones is then processed and converted in the brain in ways that we don't have to talk about, or have time to talk about. Now, the interesting thing about this example, and the reason I brought Dalton's story up, comes from a study done actually only about a decade ago by David Hunt and his colleagues who were at the University College in London. It turns out that Hunt realized that Dalton's eyes had been saved. They had been preserved by the Manchester Literary and Philosophical Society for literally 150 years. And so what Hunt did was to ask if he could account for Dalton's color blindness because of a mutation in a particular opsin gene. So Hunt and his colleagues got permission to take a small piece of tissue from Dalton's eyes that had been preserved and to extract the DNA from that. And then using molecular techniques, they were able to compare the gene sequences of the opsin genes that Dalton had with the normal opsin genes of a normal vision person. And indeed, they found out that Dalton did have a mutation in the gene coding for the opsin associated with receptors specifically sensitive to the color green. I think this story provides a really interesting and fun and dramatic illustration of the fact that sensory tuning in the visual modality for different wavelengths or colors depends very exquisitely on the molecular structure of the receptor molecule itself. And it also provides an interesting demonstration of the power of modern genetic technology to be able to unravel this puzzle about Dalton 150 years later using these kinds of techniques. Well, clearly, differences in sensory modalities and tuning within those modalities represent only the most fundamental properties of a sensory stimulus. Of course, sensory systems have to be able to detect and process many more specific features of the external world in order to account for the richness of sensory experience that we have. Much of this richness, certainly the richness of our own human sensory experience, comes not from the sensory receptors directly, but instead is due to processing in the brain. Now, this is something that we only began to touch on last time when we talked about synaptic integration, and of course it could serve as the subject of its own course. But the sensory mechanisms that we have been able to look at illustrate how sensory experience must, at some level, depend on molecular specializations of cells, which initiate a complex cascade of processing that extracts more and more specificity from information available in the environment at increasingly higher levels as we move up in complex nervous systems.